So we're moving into the final system, the hematologic system. The last two lectures, today and next time, will finish up the course. So I would strongly suggest that you, if you haven't already started, start studying for the final exam now. Don't wait until the last week. There is so much that we've covered that you'll just be going through questions and questions and questions and questions forever, and it may kind of blow your mind. So, you know, start every day when you do some studying, go over a couple of sets of questions from earlier in the semester. They'll seem like they were forever ago, but they'll also, you know, they'll come back to you pretty quickly. And that way, as you approach the thing, you can blaze through them one more time maybe, but you won't have to do so much to prepare for the final. So I'm going to do a bit of a review on this because those of you who've already had A&P 1 and 2 have had a pretty good covering of the hematologic system. However, those of you who are taking A&P 2 right now at the same time, you're probably just having it right now. That should be your last system in A&P 2 as well as this one. So I want to make sure you understand what's going on while I talk about some of this patho stuff. So your blood is made up of a couple of different components. The plasma is clear extracellular fluid. It's not inside the cells, but the red blood cells make up a good portion of your blood. The plasma, it's about 60% of your blood. Red blood cells make up about 40%, depending on what your concentration is of those red blood cells. And plasma itself is 90% water. About 10% is actually things dissolved in the plasma. Electrolytes, plasma proteins, and the like. So you have your plasma, and then you have the formed elements, which are your red, white, and white blood cells, and thrombocytes, which are platelets for clotting. Erythro means red, red blood cells. Leuco means white, white blood cells. Thrombo means clotting, clotting blood cells. And the blood has a lot of functions. It's a connective tissue. It connects all parts of your body one to the other. It delivers oxygen, removes carbon dioxide and other wastes, takes the waste to the kidneys to be eliminated. It is your immune system is carried through the lymphatics and the regular blood vessels. So all of these white blood cells are part of the immune system. Your clot formation is controlled by the blood. So if you have a bleed in there, it takes care of that. It carries hormones from one part of the body to the other to signal that part to do what this part says to do. Maintains your acid-base balance. You either pee out or maintain more hydrogens or more bicarbonate, and therefore keep your pH at 7.4 where it's supposed to be. And it's involved in thermoregulation. Takes blood to the surface so you can sweat and cool yourself off, or retracts the blood away from the surface when you need to retain heat because it's cold outside. Also dissolved in the plasma and somewhat within the blood cells themselves, your electrolytes. Sodium, chloride, potassium, calcium, and bicarbonate are your main ones. Your plasma proteins are big protein substances that are there for a couple of reasons. One is to add intracellular osmolarity to help pull, to keep your blood pressure from pushing all the fluid out into the tissues. So the capillary oncotic pressures or osmotic pressures are maintained partly by those plasma proteins. And various albumins and globulins act as transporters. They transport your steroid hormones. Any of your lipid hormones, like thyroid hormone, have to be carried in the plasma that way. Globulins can be antibodies, your immunoglobulins, your antibodies, and it carries clotting factors. The red blood cells are little flat biconcave discs. They look like a donut that the hole didn't go all the way through. They're very deformable. They have to be able to bend to actually fit through a capillary. They don't have a nucleus. They lose the nucleus in their formation, and it makes them bendy. Makes it easier for them to bend. And they have no mitochondria. They can't use oxygen to make energy. All they can use is glycolysis. They're very simple. It makes them easy to bend. It also makes them not last or live very long. They can't really replicate any of their own parts. And when they start wearing out, they break apart and they get filtered out by the liver and the spleen out of the system, which gets rid of the parts of the red blood cells that you don't want, like the bilirubin goes in the bile and gets pooped out, and the things that you want to maintain, like iron and some proteins and things, are retained and recycled. About 120-day lifespan. You make billions of them every day. They have hemoglobin, which transport oxygen to the tissues and carbon dioxide back. Your hematocrit is the part of your blood which is actually red blood cells. It's somewhere around 40%, a little bit less in women, a little bit more in men. And production of red blood cells is stimulated by erythropoietin. Now, this is something that athletes take when they want to cheat sometimes because it makes more red blood cells so they can carry more oxygen. It's illegal. But erythropoietin is produced in the kidney when you have low oxygen content, and it causes your bone marrow to make more red blood cells. Platelets are smaller than red blood cells, little guys, little flat discs that clump together and form the first part of a blood clot formation. And they are stimulated by thrombopoietin. Poiesis is the creation of something. So thrombopoietin, the liver makes thrombopoietin and causes your bone marrow to make more thrombocytes, platelets. Your lymphatic system, if you'll remember, is the cleaning system of the tissues. You have lymphatic vessels going in the same place you have capillary beds. It pulls out the debris, it pulls out extra fluid, and it pulls out bigger things like bacteria, cancer cells, things that can't be easily moved into a capillary. It pulls them into these big fenestrated holes in the lymphatic blood vessel wall, brings that lymph slowly back up to the lymph nodes, which are chock full of lymphocytes, white blood cells that are designed to kill the bad guys. And so it supposedly sterilizes this fluid before it releases it back into the circulation. The spleen is a large, giant lymph node kind of tissue. It also has the job of removing some dead red blood cells from the circulation. Hematopoiesis means creating blood cells. And what you have in your bone marrow are stem cells. Pluripotent means they can go different ways. They're potent to move in different directions. And depending on whether you stimulate with erythropoiesis or thrombopoiesis, or stimulatory cytokines from the lymphatic system wanting more white blood cells, they morph into a red blood cell, white blood cell, platelet. That's where they're finding stem cells that they're using for stem cell studies nowadays. Bone marrow is called myeloid tissue. Myelo it means the center of, and therefore myeloid or myelose can mean bone marrow. The red bone marrow, which makes blood cells, is mostly found in flat bones and some irregular bones, so your skull, your sternum, your hips. If they take a bone marrow transplant for blood cell production, they usually take it out of your hip bone, out of the iliac bone. Yellow bone marrow is found a lot in the long bones, in the shaft of long bones. Erythropoiesis starts out with a big nucleated pluripotent stem cell. It is committed to become a red blood cell, and it starts conversion in that direction. It loses size because it's going to have to fit through capillaries, and it loses its nucleus. It's chock full of hemoglobin, which is produced from iron or with iron. Regulation: when oxygen drops, the kidneys make more erythropoietin. It goes to the bone marrow. They always draw the wrong kind of bone. This is not the kind of bone that makes much red blood cells, but everybody recognizes that as a bone. It makes a bunch of red blood cells. They pick up a lot of additional oxygen, and the negative feedback is the increased oxygen level. Hemoglobin is a very complicated molecule. It has 4 hemes, which have an iron center, which gives them their red color. And each of these hemes can carry 1 molecule of oxygen. So your hemoglobin is designed to grab oxygen at the lungs, release it at the tissues. It also picks up CO2 when you're down at the tissues and you get rid of the oxygen. The CO2 can bind to binding sites on the globin parts of the hemoglobin. Iron is required for the production of hemoglobin. If you don't have iron, you don't make enough hemoglobin, and the hemoglobin helps actually determine the creation of the red blood cell and even the shape of it. So, we eat iron, we absorb iron in the gut. It is stored in the liver. Excess iron should be secreted out with the bile, except we're not very good at that because historically, evolutionarily, people never had too much iron. They always had too little. We got really good at storing it, but really bad at excreting excess iron. So it's possible to have iron excess that will damage tissues, and especially the liver. The iron then is transported in the blood by a substance called transferrin. So it comes out of the liver, binds to transferrin, it goes to the bone marrow to make erythrocytes, and then the erythrocytes are filled with hemoglobin full of iron. When they break down, the hemoglobin and the iron is recycled, and the process starts over again. Hemostasis, stasis means stopping. This means stopping blood clotting. blood bleeding, stopping bleeding. So it's forming a blood clot to stop the bleeding. So the first thing we need is platelets. You have to have enough thrombocytes to be able to form the platelet plug in the first place. And then we have clotting factors that will cause a true blood clot to form. The platelet plug is like Jell-O. It's better than nothing. If you cut yourself and it stops pretty quickly, that's the platelets. It takes a little longer than that for a blood clot to actually form. And then the clot will stay there long enough for new endothelium to grow back so that it's healed, and then the clot gets dissolved by a TPA, which is a substance that we make in our bodies that dissolves clots away. So here we have, you get a damage, your endothelial layer releases tissue factor, tissue activating factors, and they cause the platelets to get all spiky and stick to each other and form the platelet plug. As this happens, you start sending out cytokines that ask for the clotting factors to arrive. The clotting factors will ultimately produce fibrin, which is the substance that holds the clot together. And it requires quite a cascade of events to form this fibrin clot. The fibrin fibers then squeeze together, squeeze out the juice, and the clot becomes harder and harder till it's more like a scab. Then at the same time, you're producing plasmin, which degrades the fibrin and causes the clot to go away. So it should stay just long enough and not too long. Plus, you're always creating plasmin out here so that the clot won't grow any bigger than it needs to. It's restricted to the site of the damage. Platelet plug adhesion requires von Willebrand factor and tissue factor. If you're deficient in those, you can't start this process properly. They activate and get spiky, they aggregate. Then the clotting factors start showing up. They come from two places. Intrinsic factors are already there in the wall of the blood vessel. The extrinsic factors have to come from outside, from the blood itself. So you have an intrinsic cascade and an extrinsic cascade. They combine forces and form a blood clot. This is designed as a protection against forming clots all the time. You have to activate the platelets. You have to ask for these two guys to get together, and then you form a clot. Otherwise, we form clots and kill ourselves all the time. Coagulation cascade, platelets first. The extrinsic clotting factors come from the blood. The intrinsic ones are already there in the tissue. Prothrombin becomes thrombin. Thrombin causes fibrinogen to become fibrin. So it's a cascade of events. The fibrin then produces the actual blood clot. Clot retraction turns into a scab. And then TPA. Now you may have heard of TPA. This is what we give people who've had a stroke to break the blood clot. Or if they have a pulmonary embolism, you shoot TPA in there, it dissolves the clot. The moment we start making a clot, we start breaking it down. We start producing TPA to keep it from spreading and blocking off the blood vessel. TPA, there's other activating factors too, but tissue plasminogen activator activates plasminogen, the creator of plasmin. Plasmin then breaks the clot apart. How do you evaluate to see if your system is okay? Well, you can take out some bone marrow and look at it. examine it under a microscope to see if it has abnormal cell development. You can measure the iron stores and other factors in the bone marrow. You can do a differential cell count to see how many red blood cells you have compared to neutrophils, eosinophils, basophils, and a lot of blood tests. You can do, prothrombin time. You can do a thrombin time test. The prothrombin time really tests to see if the extrinsic factors are okay, because once you already have thrombin, all of that can happen internally. And so thrombin time, you add thrombin and you see how quick it clots. And if it's delayed there, then you know it's an intrinsic factor problem. If it's prothrombin, if you add prothrombin and it's delayed, then you know there's a problem out with the extrinsic pathway. Couple things about kids, neonates especially. They tend to have too many blood cells early on for a couple of reasons. There is stress involved with being born, and it activates a lot of their mechanisms that stimulate the blood cell formation. Plus, there actually is a certain hypoxia to their environment in the mother. They're not breathing. They're getting enough oxygen to grow, but it's not any more than they need, and as a result, they make a little more erythropoietin. They make a few more red blood cells, and so they're likely to be born with polycythemia that will last a short amount of time. The red blood cells will die, and they won't make new ones as fast until they get to about the level they need to be. Kids also tend to have more lymphocyte counts because they're always getting infections. Their immune systems aren't that great yet. They're always getting infections and colds and croup and ear infections and stuff, so their white blood cell counts are elevated compared to adults to some degree. Okay, so that's just the background on the hematologic system. If you already had that, sorry to put you through that, but hey, you can always fast forward if you need to, right? Let's look at some problems that happen with the blood system. Anemias are quite common. What it really means is no blood. Well, that isn't actually a very accurate term for it. What it means is you have insufficient functioning red blood cells. They could be shaped wrong. They can lack hemoglobin. You might not have enough of them. They might be too big to fit through the capillaries. So there's a number of things that can go wrong with the red blood cells to give you anemia. Don't make enough of them. Blood loss, if you're bleeding internally, you can be anemic because you aren't keeping enough blood in your blood. They may be breaking down too early. You could have hemolysis, or some combination. So we can look at anemias in a couple of different ways. What caused them? Iron deficiency anemia. That's an easy one. Morphology. What's wrong with it? Are they too big? Are they too small? Do they have too much iron in them or too little hemoglobin? So the morphology is based on abnormal size or hemoglobin content. They can be too big, too little, or normal sized, and that size cytic cell size, macrocytic, microcytic, normocytic. Or they can have hemoglobin content that's either normal or too low. Not really too much hemoglobin is common, although you can have too much iron. But normochromic, chrome means color, and hemoglobin makes blood red. So normochromic, hypochromic means they're kind of faded out. They don't have enough hemoglobin. Or they can be shaped funny, like sickle cell anemia. Iso is a term that means the same. So isocytosis means all the cells look the same. Okay, look the same shape. Anisocytosis means they're not. Means you've got big cells, little cells, funny shape, whatever. Poikilocytosis means they're different shapes. So they may not be too big or too small exactly, but they may be sickled or they may be square or something. So you can have ones that are either they're all the same size or they're not. They're all the same shape or they're all not. And so what really happens ultimately is you don't transport oxygen well enough. If you don't have enough hemoglobin, if you don't have enough cells, if the cells are the wrong size, they can't go through blood vessels, one way or another, you're not carrying enough oxygen. Hypoxemia means not enough oxygen in the blood. Hypoxia means you don't have enough in the tissues. Again, it could be because you don't have enough in the blood, or maybe you blocked it off and the blood can't get there. But hypoxemia means you're not carrying enough in the blood. Get tired, weak, can't breathe very well, you're always gasping for air because you don't have enough oxygen. Get pale because hemoglobin is red, it makes you pink. If you don't have enough oxygen, you get kind of pale. Let's look at a couple of these anemias. What if you're macrocytic but normochromic? Well, that means you have some cells that are too big, but they have their hemoglobin is okay. The big problem with this is they don't fit through capillaries, so you can't get the oxygen delivery to the tissues that you need. Megaloblastic anemia is another term for it, and they're almost entirely two types. One is vitamin B12 deficiency and the other is folate deficiency. It's really interesting because B12 almost entirely comes from animals, folate almost entirely comes from plants, and therefore it's a proof that we were evolved to be omnivores because you get the same type of anemia if you're short of this one or you're short of that one. The B12 deficiency is called Pernicious Anemia. They called it that because they knew what iron deficiency anemia was, but they gave these people iron and they were still anemic. It was pernicious. They couldn't get rid of it. It's the most common one. It can be because you don't eat enough B12. If you've become a vegan and you're not careful about getting the vitamins that you normally get from meat, then you can develop a B12 deficiency. Most B12 deficiency, at least in Western countries, is because your stomach lining doesn't make intrinsic factor, or not enough of it. B12 has to bind to intrinsic factor and get absorbed in the ileum along with intrinsic factor. Pernicious anemia, megaloblastic. Look at the size of some of these red blood cells. Blocks off blood vessels, tongue gets really red, rashy around it, loss of appetite, beefy red tongue, jaundice, because these large blood cells block some of the tubules in the liver. Enlarged spleen, same reason. Neurologic damage, it's lethal. People died from pernicious anemia. Absence of intrinsic factor usually. And taking vitamin B12 supplements may not do it. Because if you don't have any intrinsic factor, you won't absorb that either. You may need B12 shots. Folate deficiency anemia produces the same megaloblastic pernicious anemia effects. All you have to do is take some folate. It's not something that you're not making inside, you're just not getting enough folate. Microcytic hypochromic. So now the cells are small and don't have enough iron in them. You don't have enough hemoglobin in them. Hypochromic, not enough hemoglobin in them. This is a white blood cell here. But look at these little kind of shriveled white, red blood cells. They're really pale. They don't have enough hemoglobin in them. Well, you either disorder of iron metabolism, you can't use the iron properly to make hemoglobin. Iron deficiencies, you don't eat enough. Iron deficiency anemia is the most common microcytic hypochromic anemia, although there are other things. You can actually have anemia from an insufficiency of copper and a couple of other micronutrients. Worldwide, most common type of anemia, iron deficiency. You don't need much iron, but you need some. For years, for millennia, people got enough iron by cooking in iron skillets. I worked for a health organization, Centers for Health Promotion in Atlanta, and it was kind of a global idea thing, a small group trying to do monster things. Well, one thing that we actually did accomplish is our president flew to China and convinced them to let him provide iron supplements to put in soy sauce and rice flour, and basically wiped out iron deficiency in a fifth of the world's population all at once. It was kind of a big idea. Nutritional iron deficiency, metabolic problems dealing with it. When your iron drops to the point where your hemoglobin drops down to about half what it's supposed to be, you are in hemoglobin deficiency, probably iron deficiency. Normal hemoglobin for men is about 16 grams for every deciliter of blood. Women, it's about 14. If it drops to half of that, you are in deficiency. Couple of iron deficiency anemia problems. This kind of looks like the pernicious anemia tongue. This inflamed tongue and the inflammations in the corner of the mouth. Koilonychia means oddly shaped nail condition. Onycho is the term for nail. It kind of cups and scoops up. So if you have really pale conjunctiva, sores around your mouth, scooping thumbnails, take some iron. Normocytic, normochromic. So the cell's the right size and it has the right amount of hemoglobin. So what could be the problem? You don't have enough of them. You're either not making enough or they're breaking apart too soon, or you're bleeding them out. Okay? But there's nothing wrong with the cells themselves. For some reason, you just don't have enough of them. Aplastic anemia means you're not making them. Pancytopenia, neutropenia being the term for not enough. Your bone marrow isn't making enough of any kind of blood cells. Or maybe you're just not making enough red blood cells. Maybe your kidneys aren't making enough erythropoietin. Post-hemorrhagic anemia, you bleed half your blood out, you don't have enough blood. So that's an easy one. Hemolytic anemia means they're breaking apart. For some reason, the cell membranes are weak, and the red blood cells wind up lysing, hemolysis. Or they're agglutinating, they're clumping together. These are just as bad as not having any. Actually, they're worse, because now this clump can't fit through a capillary. It may block off the capillary. But these are no longer usable red blood cells. So you only have a few that are really working very well. Could be a couple of coagulation type abnormalities, or particularly if you got a blood transfusion of the wrong blood type. It causes agglutination of your red blood cells. Warm agglutination is worse during fever. It agglutinates when your body temperature goes up. Cold agglutination, it causes your blood cells to clump together if your temperature goes down. Sickle cell anemia. Inherited. This is a normal red blood cell. These have sickled. They actually have a different kind of hemoglobin. Remember I said hemoglobin helps determine the shape of the cell. Hemoglobin S is hemoglobin sickle cell. Okay? So hemoglobin S will cause the cells to sickle under certain conditions. Low oxygen causes them to sickle. Somebody may live in Miami and never know they have sickle cell anemia. They come to Taos to go skiing and they have an episode of sickle cell anemia. It's almost entirely exclusive to people of African descent. It actually provides some protection against malaria, and therefore they lived at low altitudes. It didn't give them the problems it gives people at higher altitudes, and it protected them against malaria. But now they come to the U.S. and they go up into the Appalachians for a summer vacation, and they have sickle cell and, a variety of problems. Here's a cell here that has sickled. And not only this can't carry oxygen very well, it doesn't bend and go through capillaries properly. So a lot of the sickle cell problem is blocking of blood flow through the capillaries. Chronic inflammation, if you have lupus, if you have cancer, hepatitis, AIDS, those things will affect your bone marrow's ability to utilize iron to make hemoglobin. It'll affect the ability of the red blood cells to do their job. It may cause them to break down quicker. And as a result, these conditions are often accompanied by anemia. Now, what about the other side of it? Polycythemia. Remember I said the baby who was just born and was in kind of a hypoxic womb produced more erythropoietin, made too many red blood cells, and by adult standards actually had an overabundance of red blood cells. You can have polycythemia because you make too many red blood cells. You can have a tumor that overproduces whatever tumors do. They're overgrowth of cells. If those cells are red blood producers, you might produce too many that way. You can have a relative polycythemia if you get really dehydrated. You lose water, and now all the cells are packed together too much. Your hematocrit gets so high that it makes it hard for your heart to pump the blood through your blood vessels. Couple of different types of absolute polycythemia, which means that you haven't lost water, you're just making too many red blood cells, caused by the bone marrow. Either there's the bone marrow stem cells are overactive and convert into red blood cells too easily, or cancer, a cancerous tumor of the bone that causes an overgrowth of those cells and they make red blood cells like mad. Secondary is you're responding inappropriately to erythropoietin. Either you're making too much of it or you're responding too much of it to it, and therefore you're making too many red blood cells. Buddy of mine played on the 60 and over, I guess it was, world champion rugby team a couple of times. And they always like to play their tournament in Aspen, Colorado. Well, that's really cool, except here's a bunch of 60-year-old guys going up to high altitude, and a lot of them live in Florida and places like that. So they go up there and they huff and puff because there's no oxygen up there. And their kidneys make a lot more erythropoietin because they respond to the low oxygen, and they make more red blood cells. And by the end of the tournament, they're actually responding better. They're feeling a little bit better. It's why people do high altitude training before events. They go to altitude, they make more red blood cells. But he came home and he had a doctor's appointment for a normal physical, and the doctor took his blood and said he had polycythemia. He was going to have to, every month for the rest of his life, he was going to have blood taken out and maybe fluid put in and, keep the blood thin. And he said, Doc, I just went to Aspen for a couple of weeks and, I've made more red blood cells. And the doctor said, no, that couldn't happen that quick. It does. He went back a month later and his blood count was normal. Iron overload causes hemochromatosis. In the blood, it's too colorful condition. Hemochromatosis, very bright, lots and lots of iron in there, and it's toxic. Toxic levels of iron can destroy your liver, destroy your spleen, cause nervous system damage. And the problem is that most hemochromatosis, you can take too much iron supplementation and give yourself hemochromatosis. It has been documented to happen. But normally it's this hereditary hemochromatosis. You just don't eliminate any. And you always eat a little and it builds up and builds up and builds up over time. And next thing you know, you've got too much iron. And you really do have to bleed the person. Take some blood out and replace it with fluid, with clear fluid, to thin all this out. Hematologic 1.